So in the last video, we talked about micro and macroevolution. We talked about reproductive isolation leading to speciation. And in the remaining lecture, the second lecture, we will talk about other ways in which speciation can occur. Two common mechanisms of speciation are allopatric and sympatric. So allopatric is when you have a physical barrier separating two species or two groups of individuals leading to the formation of a new species. In sympatric, you actually do not have geographical isolation, but you still end up with two new species or one new species from the old species. So here is our original population. We have drought or some reason that the water levels go down, have a physical separation, and end up with this new species from our original population. Here we just have a spontaneous formation of a new species from our original species without any type of geographic isolation. So allopatric or other country speciation is where gene flow is interrupted when a population is divided geographically, as we just showed in the last picture. And so geographic barriers can be a canyon even. So the Grand Canyon can create a barrier for small rodents, but not birds. So the rodents can't get across the canyon, but the birds can fly across. Coyotes are trees with windblown pollen, because the coyotes are more mobile. Trees with the windblown pollen can all still travel across that canyon, but the small rodents cannot. So these are geographical barriers, even if you don't see a visual barrier, because of the size of the animal or its means of locomotion, may eventually lead to some type of geographic isolation. You can also have predation. So the mosquito fish in the Bahamas, several isolated populations in different ponds. Some of those ponds have lots of predation, others do not. And so with the ponds that have predatory fishes, head is streamlined so they can swim faster. Tail is powerful, enabling the rapid burst to escape predators. Where there are ponds without predation, they have different body shape that favors long, steady swimming, so kind of like a marathon runner. And so when you get separated in different bodies of water, depending on what else lives in there with you, you can also lead to speciation. You can even reproduce allopatric populations in the lab. So we took fruit flies, raised them on either starch medium or maltose medium, and then we took those two groups after they evolved from 40 generational matings on the same medium. And what they found is that females raised on starch and males raised on starch mated 22 times. But if you took the females raised on maltose and mated them with the male starch, only 9. Same thing with female starch, male maltose, 8, but took the male maltose and the female maltose, 20. And so here you can see that they develop an evolutionary preference for organisms that are raised on the same medium. And just to make sure it wasn't just because they were in two different populations, so you have population 1 starch, population 2 starch, male and female, and whether you mix them with population 1 or population 2, you don't see a significant difference in the number of matings. So these guys, allopatric separation due to the bottles they were in with the medium they were raised on, adapted to that food source and found a preference for another organism raised on the same food source. Here are the shrimp. And so in nature, the shrimp were separated when the Isthmus of Panama developed. And this was 9 to 3 million years ago. They became isolated and formed unique species of snapping shrimp. And so you have 2 on this side of the isthmus and 2 on this side of the isthmus that have diverged into specific unique species. And so what is the evidence of allopatric speciation? We've seen naturally and in artificial that isolated populations or highly subdivided typically have more species than no regions with few barriers. Isolation between populations usually increases as the geographic distance between them increases. Geographic isolation does not prevent interbreeding. Physical separation is not a biological barrier to reproduction, it's just a geographic barrier. Biological reproductive barriers are intrinsic to the organisms themselves, and so even if you put them together, they can't breed. No interbreeding. And only biological barriers can prevent interbreeding when populations come into contact with one another once that geographic barrier would be removed. So Sympatric, same country. We have several examples when gene flow is reduced due to polyploidy, so the number of chromosomes, habitat differentiation, and sexual selection. And so polyploidy is when you have extra sets of chromosomes, and those extra sets of chromosomes lead to the inability to form offspring. Animals are more prone to be affected by this. Plants usually enjoy polyploidy. And so occasionally occurs in animals, but not normally. It's usually lethal. Much more common in plants. Find it in many important crops: oats, cotton, potatoes, tobacco, wheat. And so wheat used for bread is a allohexaploidy, 6N. And then plant genetics produce new polyploidies using chemicals to induce errors in cell division, but it doesn't tend to affect the plants. And so here's a quick video talking about the polyploidy.
Let's use this simplified cell, which represents a diploid plant cell, to examine how changes in a cell's ploidy can contribute to the formation of new plant species. This cell has 2 sets of chromosomes, or a ploidy equaling 2N, each set consisting of 1 red and 1 blue chromosome. In a pair of homologous chromosomes, 1 chromosome originates from the female parent, indicated by the stippling, and the other originates from the male parent. When a diploid cell undergoes meiosis, it normally produces 4 haploid daughter cells, each with 1 set of chromosomes, or a ploidy equaling N. Every so often, an error called non-disjunction occurs in meiosis, and some daughter cells get too many chromosomes, while others get too few. Non-disjunction can occur in meiosis 1 or meiosis 2, but here we'll look at an example in meiosis 1. In meiosis 1, chromosomes would normally segregate from each other, with the chromosomes of each homologous pair going to opposite poles. However, in this case, all the chromosomes migrate to one pole. One daughter cell has no chromosomes, and the other has 4. The cell with 4 chromosomes continues through meiosis 2. The chromosomes line up at the middle of the cell, and the sister chromatids separate. The cell divides, producing 2 cells. Through non-disjunction, a diploid parent cell has produced 2 diploid gametes. In contrast, normal meiosis results in 4 haploid gametes. They're called gametes here for simplicity, but keep in mind that in plants, the production of gametes requires many more steps. What would happen if 2 diploid gametes came together during fertilization? When 2 diploid gametes fuse during fertilization, the resulting zygote is a polyploid cell, meaning that it has more than 2 sets of chromosomes. A cell with 4 sets, or a ploidy equaling 4n, is more specifically called a tetraploid. This cell can also be called an autopolyploid because the doubled chromosomes are from the same species. The prefix auto means self. Polyploid plants survive perfectly well, but they cannot produce fertile offspring when mating with their diploid counterparts. To see why, we'll observe several rounds of meiosis and fertilization. First, let's see how a tetraploid undergoes meiosis. Like all cells, it first replicates its DNA. During meiosis, the homologous chromosomes may come together in pairs, in 4s, or in other arrangements. The cell divides during meiosis 1. It divides again during meiosis 2. The result is 4 diploid gametes. Let's now consider the act of fertilization. When 2 normal haploid gametes fuse, the result is a normal diploid zygote. If 2 of these diploid gametes came together in fertilization, the result would be another tetraploid zygote that would continue the generations of tetraploids. However, when one of these gametes fertilizes a haploid gamete from a normal diploid plant, the resulting zygote has 3 sets of chromosomes, or a ploidy equaling 3n. It is triploid. Although a triploid zygote typically develops into a viable plant, meiosis in the triploid is a confusing affair. Before meiosis, the cell replicates its DNA. During meiosis 1, the homologous chromosomes undergo synapsis. The extra chromosomes lack homologs. They may pair with the other 2, or they may not. The cell divides. Each daughter cell receives a unique assortment of chromosomes. The cells divide again during meiosis 2. In this case of triploidy, all the gametes have an extra chromosome and are designated N plus 1. In a system with more chromosomes than this example, every gamete has a different assortment of extra chromosomes. Apparently, this imbalance prevents seeds and pollen from developing normally. Many so-called seedless cultivars, such as bananas and watermelon, are triploids. Tetraploid gametes, on the other hand, have a balanced number of chromosomes. Because the tetraploid and diploid plants mate to produce sterile triploids, they are reproductively isolated and are considered 2 separate species. These diploid cells are from 2 different plant species. If the plants mate and create hybrid offspring, speciation can result. One of the cells has a diploid number of 4 chromosomes, and the other has a diploid number of 6. When these cells undergo meiosis, they produce cells with haploid numbers of 2 and 3 chromosomes, respectively. Let's say that pollen from one species fertilizes an ovule from the other. In this case, the hybrid zygote may be viable because the sets of chromosomes it receives from each parent are complete. However, because the chromosomes don't exist in matching pairs, this zygote is essentially haploid, with a haploid number of 5. Without pairing chromosomes, a cell cannot divide by meiosis, but it can divide by mitosis. If a non-disjunction error occurs in mitosis, the cell's ability to divide by meiosis can be restored. Before mitosis occurs, the cell replicates its DNA so that each chromosome consists of 2 chromatids. The chromosomes move to the middle of the cell. In this case of non-disjunction, all of the chromosomes move to one pole of the cell as the cell divides. The daughter cell that receives the chromosomes is now diploid, and it has a diploid number of 10. A cell with pairs of chromosomes can divide by meiosis, producing gametes with 5 chromosomes. These gametes can fertilize each other, resulting in a diploid zygote with 10 chromosomes. This zygote represents the beginning of a new species. It contains all the chromosomes from its 2 parent species, but is now reproductively isolated from its parents because they all have different numbers of chromosomes. An individual of species C is an example of an allopolyploid, which is formed by combining gametes containing chromosomes of two distinct sets, followed by an error, such as non-disjunction, that results in chromosome doubling. Speciation based on allopolyploidy is common in wild plant species.
So there's your polyploidy. You don't have any possible exam questions really on this, but you will see some of the multiple choice. So audiploidy, individuals with more than 2 chromosome sets derived from one species. And then as we just saw, the allopolyploidy, or when you have parents derived from 2 separate species. And then once that new generation comes about, they can mate and make new offspring, but they can't interbreed with their original parent species. So that's the difference between allopolyploidy and autopolyploidy. At least 5 new plant species have evolved by polyploidy speciation since 1850. And so here is our 3 diploid species of goat's beard plant, Traganopogon. They have interbred to produce 2 new tetraploid species from the original T. dubius. And then these new offspring actually have 12 also. And the last way in which we can get sympatric speciation is through new ecological niches. So in North America, the apple maggot fly lives on native hawthorn trees as well as more recently introduced apple trees. And then the flies use different host species or reproductively isolated, both habitat and temporal isolation. So time is temporal, habitat is where they actually live. Also with our cichlids, which is a fish, we've had sympatric speciation with sexual selection. So in normal light, this species is more colorful than this species, but if you put them under a monochromatic orange light, you really can't tell the difference in them. And so the different colorations in normal light probably contributed to the speciation in the fish in Lake Victoria. And this would be a morphological preference. And another example of a sexual are our peacocks. So the great displays of our male peacock attracting the female. So here is a review of allopatric and sympatric speciation, going over why they lead to speciation. Remember, sympatric, result of polyploidy, natural selection, or sexual selection. Whereas our allopatric speciation, geographical isolation, restricting the gene flow.
Two groups of animals are defined as different species when individuals from one group don't mate and reproduce with those from the other.
The 150 species derived from one.
One way a species can split into two is for populations to separate geographically. Over many generations, they can undergo enough changes in their respective habitats that if and when they come back together again, they don't mate.
Now, suppose that a population of these lizards ended up in an area that was much more open and sunnier. In that case, a light-colored dewlap isn't very effective. So over time, the population would evolve by natural selection to have darker dewlaps.
If for some reason these two populations come together, the females would no longer recognize the males as members of the same species. They wouldn't mate. They would be reproductively isolated. Once new species have formed, competition drives the evolution of different body types. Species living in the same area compete for resources. But if members of one species move into another habitat, they can use resources not available to the other species. Over many generations, natural selection favors traits that enable species to occupy different habitats.
And here are our last two models of tempo speciation. So we looked at micro and macro before. And so another way to look at that is punctuated. So new species change most as they branch from a parent species. So a quick change, this is punctuated. Whereas gradual is you have a very slow change over a steady time that leads to two new species rather than one punctuated change that leads to the new species suddenly. So very similar to our macro, which would be similar to punctuated, or micro, which would be similar to the gradual model. So these are models, not mechanisms. All right, so that is all of chapter 22. I will hopefully get out an exam review video tomorrow. But if you have questions before then, be sure to shoot me an email and I will get back to you. Have a great one.